The Human Genome Project, or HGP, was designed for sequencing the human genome. In 1990, the HGP officially began as coordinated research from France, Germany, Japan, China, and United Kingdom. However, it was not until 2001 that the HGP International Consortium published the first draft of the human genome. The HGP researchers deciphered the human genome in three significant ways. One is determining the order of the chemical bases of A, T, and CG. 2. Generating maps for genes in our chromosomes. 3. Producing linkages from individualized maps. The technology used in the HGP was an improvement from previous studies. Similar to the sequencing of Phi X174, an improved version of Fred Sanger's method known as whole genome shotgun sequencing was used. A sequenced human genome needs to be transformed based on its initial observation. Scientists have partially disseminated this information in the human genome for the benefit of our species. Initially, the draft sequence revealed 85% of its content with an accuracy of 99.99%. Gaps in the genome were filled through comparative research on C. elegans and Drosophila melanogaster by identifying and aligning homologous sequences with the human genome. With the extrapolation of data on other databases, the first draft of the human genome yielded 2.85 billion base pairs. After determining the order of chemical bases in the human genome, scientists constructively compared these results with other species. For example, C. elegans yielded 100 million base pairs. Drosophila melanogaster has 180 million base pairs. S. cerevisiae contains 12 million base pairs. And lastly, Amoeba dubia contains 670 billion base pairs, which is 200 times more than the human genome. Based on this comparison, if the complexity of a species is dependent on genome length, then Amoeba dubia, a single-celled eukaryote, is a more progressive organism than humans. However, it is not the case. To determine what complexity meant, scientists looked at the number of genes in the human genome. Researchers found that the human genome contains 30,000 to 40,000 genes, significantly lower than the estimate of 100,000 genes. Comparatively, C. elegans were found to have 20,000 genes, Drosophila melanogaster with 14,000 genes. Unfortunately, the number of genes in Amoeba dubia remains unknown. The human draft sequence revealed that coding and non-coding regions are concentrated randomly on areas of a genome. Gene-rich regions are predominantly GC, while AT are concentrated on gene-poor regions. Results showed that only 2% of the human genome encodes for the protein coding gene. This led to the investigation of alternative splicing. The prevalence of alternative splicing was analyzed by constructing mRNA transcripts which cover coding regions of chromosome 22. The chromosome generated 642 transcripts for 245 genes, averaging 2.6 distinct transcripts per gene. Comparatively, alternative splicing for C. elegans is 22%, with an average of 1.34 transcript per gene. The non-coding DNA regions are made up of repeated sequences which are mostly derived from transposons. According to the draft genome sequence, the human genome contains 45% transposable elements. In contrast, C. elegans contains 7%, while Drosophila melanogaster has 3%. Scientists hypothesized that the Homo sapiens sapiens survival against other human species relied heavily on ancient transposons. Researchers identified 20 functional human genes that are likely to be transposon-derived. However, it requires further analysis. Based on the data and observations on the human genome draft sequence, researchers were able to define what complexity meant for a species. Number 1, as previously shown, genome length is not an indication of complexity. 2. The rate of alternative splicing. The higher it is, the more complex the organism. 3. The number of non-coding regions or transposable elements are keys of unveiling paleontological records for any organism. The conclusion of the HGP in 2003 revealed more information on the human genome. Researchers identified 3.2 billion base pairs and 20,000 to 25,000 genes. Applications of the human genome include the identification of lethal SNPs which cause genetic diseases. From Mendel's law of heredity and the diploidic nature of humans, both the draft and the final sequence of the human genome can rapidly identify candidate chromosomes, genes, and loci that contain polymorphisms. Although contemporary genomics has fully sequenced the human genome, much is yet to be explored. As T.S. Eliot said, we shall not cease from exploration, and the end of all our exploring will be to arrive where we started and know the place for the first time.